Welcome to Whisk Images, where the kitchen counter meets the lab bench. You are just in time for a special serving of food for thought. What do you think of when you hear the words recipe, ingredients, 2 layers, flavor combinations, colored sprinkles? A cake, right? And DNA, of course. Wait, what? If you wonder what DNA has to do with baking, stay tuned.
Hi, I'm Joanne Tremblay Salcido, PhD Biology, aspiring baker, desserts. We may not all have a sweet tooth for science, but we do have appetites for learning. So have a seat at the Whisk Images table for a few knowledge bites on DNA, a lifelong recipe for proteins.
DNA is made from ingredients called nucleotides, pictured here as special mini macarons. They come in 4 different flavors or bases. The 4 nucleotide bases are A for adenine, T for thymine, G for guanine, and C for cytosine. Our DNA, the human genome, consists of 3.1 billion A's, T's, G's, and C's. They are packed inside almost every cell of our body. They're not just tossed in there like colored sprinkles on a cake. Oh no, it's an elaborate design. The 3.1 billion bases are linked side by side in a certain order or genetic sequence, but not as one continuous strand. Instead, our genome is split into 23 parts called chromosomes.
The DNA in each chromosome has a partner strand, a special look-alike with a complementary DNA sequence. And the 2 strands fit together like a left and a right hand, and they're connected to each other by forming base pairs according to the following rules for complementary base pairing.
An A on one strand fits with a T on the other, and vice versa. A C on one strand fits with a G on the other, and vice versa.
Base pairing causes the 2 DNA strands to spiral together and form a double helix. This molecular shape helps lock in our genetic code. Imagine that, a 2-layer cake with locked-in freshness that lasts a lifetime.
Certain stretches of our DNA spell out the recipe for proteins our bodies make. Proteins like hormones, antibodies, and enzymes. These stretches of DNA are called genes.
Genes take up only 1% of our DNA, but like a special ingredient, that 1% makes the cake.
Our genomic DNA lives in a compartment of the cell called the nucleus, pictured here as a kitchen cutting board. Proteins are made in the part of the cell called the cytoplasm, the liquid that fills the inside of a cell.
Wait a minute, if DNA stays in the nucleus and proteins are made in the cytoplasm, how does the recipe get from one compartment to the other? Enter the messenger RNA that is, also called mRNA. mRNA essentially lifts the recipe from the DNA master in the nucleus and takes it to the cytoplasm.
In simple baking terms, making mRNA is like creating a digital file of your grandmother's recipe and saving the handwritten original. Like DNA, the ingredients for mRNA include the bases A, G, and C, but instead of T, mRNA uses U, short for uracil. mRNA is produced by rewriting a gene in DNA using these mRNA bases, and the process is called DNA transcription. The DNA that carries the gene is called the coding strand or the sense strand. Its complementary partner DNA is called the antisense strand. The mRNA grows by reading the genetic sequence on the antisense DNA strand, one base at a time, and transcribing the DNA base to its complementary mRNA base. Transcription follows rules similar to those for DNA base pairing. A T base in the antisense DNA is recorded as an A in mRNA, an A base in DNA as a U, a C base as a G, and a G base as a C for mRNA. Notice that the new mRNA has the same sequence as the gene on the coding DNA, except for the U's in mRNA in place of the T's in DNA.
mRNA transcripts of a gene are made on demand and are typically thousands of bases long. The protein made from the RNA turns out exactly according to the instructions in the coding DNA. If only my desserts turned out well every time. Let's go to the cutting board for a closer look at protein making using a sample mRNA.
We'll imagine an mRNA transcript with a recipe that is only 18 bases long. The mRNA migrates to the cytoplasm where it meets up with the building blocks for proteins called amino acids. Pictured here as a serving of brownies with 20 different toppings, one for each of the 20 different amino acids used to make protein. The process of making proteins from instructions in mRNA is called mRNA translation. 3 mRNA bases in a row equals 1 codon, and 1 codon indicates 1 amino acid, in our case, a brownie. mRNA is translated by reading 1 codon after another down the line from start to finish, no skipping steps. Our sample mRNA starts with the codon AUG, which calls for the amino acid methionine, M for short. So M gets the chain started. The next codon, GUC, calls for valine, V, which attaches to methionine. Reading on are the codons for threonine, T, then isoleucine, I, and then leucine, L. The UAG codon means stop, enough. So our chain ends with leucine, and this fresh homemade protein goes on its merry way to its job.
Our genomic DNA is the same in all our cells and lasts as long as our cells. mRNA and protein, on the other hand, differ between the cells depending on a cell's specialty. Also, they don't last long, and like desserts, they must be remade. Eclairs, churros, Nanemo bars, and more. Oh my, there goes my sweet tooth. Quick, a UAG stop codon, please.
Damage to our DNA bases can permanently change our genetic sequence if not corrected. These changes, big or small, are called mutations. Mutations don't all have the same impact.
Like typos in a recipe, their impact depends on the kind of change and where in the instructions they are. In certain cases, 1 or more typos in a single gene coding for 1 protein does cause illness. These single gene defects, as they are called, are generally inherited. There are thousands of these types of disorders, and together they affect millions of people. One example of a single gene disorder is sickle cell anemia. A typo in the recipe for hemoglobin, our body's oxygen-carrying protein in red blood cells, generates a version of the protein that turns the red cells rigid and sickle-like in shape. And this blocks blood flow through small vessels and can trigger intense pain.
Here's another tip on genetic typos. Redundancy is baked in the genetic code. There is more than 1 codon for almost every amino acid. This helps reduce the potential harm of single-base mutations. We saw in our mRNA transcript that UUG codes for leucine, but there are 5 other codons that can substitute for it. It's like substituting agave nectar or molasses or maple syrup for honey. You still get that sweet taste.
Well, that's all for this serving of food for thought. Thank you for joining me at the Whisk Images table. I hope you enjoyed these few bites of knowledge. Until next time, stir things up a bit. Following rules for base pairing complementary. Okay, stop. Okay, wait.
Yeah.
The DNA in each chromosome has a partner strand.